Welcome, welcome, welcome. Now we have Group C with us here. So plan is to go through the questions on experimental models, and we feel that it would be very helpful to discuss this together. Did you consider already, Christelle, these, or you have a bit different set of questions, but anything related?
So it's I I did not recognize because we have almost the same questions, which is a good point, in fact. Um, so this morning we have talked about the same question. So, what is the best model for transgenerational effects? So, for the biota, so for us, the answer was really there is no best model because we have complementary models, and it depends on the question that you are addressing. For example, if you are really focused on several generations and transgenerational effects over a long time, you may consider to have a species with a short life cycle, such as invertebrates, for example, or plants. I don't know, Nell, but if there are plants with a short life cycles.
There
And
are like the lemna, which is only 2.5 days, but that's then a clonal organism. Or you can have the shortest we have is Arabidopsis, which is 6 weeks, which is already, if you compare that to C. elegans, it's already long. But yeah.
And then you were speaking about clonal organisms. So for the wild species, for the different biological species, you have different reproduction strategies, and some species have a clonal reproduction. So using those species, it's useful to use them, for example, microcrustacean, which is the Daphnia, which is parthenogenetic. There are only females. So if you use them to study the genetic effects over a long period, it's useful because you have this homogeneous genome at the beginning. So you may, if you have mutations, you may have a better chance to see them because there is not a high genetic diversity. But on the other hand, it's not comparable to human because there is no sexual reproduction. But it can be sexual reproduction over harsh environmental situation, but it's not the common situation for the species. Also, other models such as C. elegans, the nematode, it's a model that is also well known for human toxicology because there has been 2 model prices for this model for discovery of apoptosis and I don't remember developmental genes. And using these models, they have a small genome and they have a small number of cells. They have 956 cells, and each cell lineage is well known. So you can, but it's a simpler model. And if you are interested in a specific mechanism, you can use it. And then you can also get advantage of all the molecular tools in this models. It's like in Arabidopsis. You may have several mutants, you may have transgenes, linked to a specific fluorochrome, so you can follow the mechanisms over a long time. So it really, for us, it really depends on the question you are addressing. Then this morning with Cisco, we discussed about what would be the best model for human. So it's something perhaps we have to discuss together. So which
Yeah, I
endpoint?
think if you look at the chemical society, what they use as model systems. You also have, for example, I know a lot of work is also for radiation exposures is done on zebrafish, and I know that that zebrafish are often taken because the neurological development of the brain of the zebrafish is very similar to what is in humans. On the other hand, for example, you have other models, planaria bijvoorbeeld, for example, which is known as a model system for stem cells. Because they have this great number of totipotent cells, so it really depends on the purpose of what you're looking for, of the aim of your research, what model you should use. I think that was a conclusion this morning, wasn't it?
So it was also one of the rationale for saying that we don't have one best model, because for the environment we want to address all the radio sensitivity of all the biodiversity in the environment. So you have, I don't remember, it's 10 to 4 differences, 10,000, a factor of 10,000 between the higher radiosensitive species and the lower radiosensitive species in terms of LD50 or EDR10. So we have a wide variety of radiosensitivity, and some of these species are maybe radio resistant, but again, it depends on the question. But what is normally considered is that vertebrate species are more sensitive than the other one. Yes, but on the other hand, the pine trees, for example, in Chernobyl, it has been shown that they are highly radiosensitive. So also the different species can be used to better understand what are the triggers of radiosensitivity in the different species. I don't know, Nell, what do you think?
By sensitivity, you mean killing, LB50?
No.
Or mutagenesis?
Yeah, no. Yes, sublethal endpoints, for example, on the reproduction, because it's the endpoint of interest here. But also in terms of knowledge for DNA repair systems, you have tardigrades that can bear, I don't know how many thousands of gray, and yes, please, Professor Nakamura.
My interest or suggestion is, first thing is immature oocytes. Question is why Russell's specific locus test did not find induced mutations in those cells. My hypothesis is those cells are hypoxic. Then to recommend any kinds of experiments, well, using mice, for example, well, it is not easy. Well, we need space, facility, and money. But maybe it is now possible to create immature oocytes in vitro. Then making them under hypoxic conditions and then irradiate, and to see if hypoxia really causes very reduced frequency of mutations in those cells. That's a kind of hypothesis testing approach. But using animal has some kinds of hurdle to laboratory people. So, wow, if I am, say, 30 years old now, I would like to do that kind of experiment. Thank you.
There was some information yesterday that guinea pigs might be more like humans in terms of oocyte, immature oocytes. Can you comment on that? Do you know more about it?
Guinea pigs are much bigger than mice. That's a big problem. And also, to my understanding, there's no inbred strain. So how is it possible to generalize the data? Because Japanese guinea pig imported, say, half a century ago may well be genetically different from European, how do I say, guinea pigs fed many generations in Europe. So guinea pig is ideal, but is not useful or suited to study many animals. Or handling is not like mice. It's more difficult. That's my point.
Yes, it's something perhaps this morning we discussed about what would be the best biological model for humans, but we discussed about the idea of the best biological model. So not thinking about a specific model, but thinking about what are we looking for in this model, and then trying to see if it exists in the nature or if we have to create it. But perhaps it's true that the argument on hypoxia, the role of hypoxia in immature oocytes would be one of the important things to test.
Liter number, so it's not number in the liter, so it's not like 8 pups, 8 newborns. It's like in human, 1, 2. Guinea pigs, is it? Okay. Sorry? I don't know. Okay. I don't know. So it was my point that if we want to think of a hypothetical animal model, which is reminiscent of human reproduction features, one of the aspects to look is that litter size. It's not like 8 in mice. It's 1 or 2. Is it reasonable? Like monkeys? Yes.
Yes, it's true that it's something that is very different between some animals and human, is that due to different reproductive strategies, a lot of species invest a lot in a high number of offspring, and rather than having a very efficient repair mechanisms in one child. So it's something that has to be considered, I guess.
You were thinking about this morning. Did you find anything for this theoretical model?
No, because it came from our discussion with Cisco before lunch, but it's something that Well, perhaps will come out after when we write the papers or something like that.
Yeah, I think we also, yeah, I don't think there is an ideal model. Um, I don't think there's an ideal model for humans nor for the environment, let alone be one that fits both purposes. And I think we might also be looking at the future, where we would try to move away from doing real animal studies more to, I don't know how far they are with organoids or things like that. Maybe that's also a very good one to see um, if we can use such thing, if you want to specifically look at the sensitivity of a tissue or an oocyte or something like that. Yeah, that's. But I don't know whether there is something like a best model. I think it's you have to choose your model as best as you can for the purpose. And I agree that when you want to look at human reproduction, that the mouse, which has an offspring of 8 small mice, is not the best one. I know, for example, I also gave that example this morning. There's a they look at chicken also as a transgenerational model system, which is quite advanced. I know a group that does that. It might be interesting to see how they do it. But yeah, that's my thoughts on the best model.
Well, that's interesting, the chicken. Of course, it's relatively easy to follow the development, I think, in the
Mhm.
egg.
Yeah, I can send you the link to the group that does that. They have a very nice paper on that, and they also have epigenetic research on it. So it would fit both the transgenerational and the epigenetics. Yeah.
And during lunch, we were discussing with Zenko Herzeg about epigenetic also, because if you want to address specifically the question of epigenetics, you need to have a model which is easy to study epigenetics. And sometimes the problem is to have stable epigenetic marks, enfin, to choose which epigenetic marks is the most stable and the most relevant for transgenerational effects. And it seems that DNA methylation and histone modifications are among them. And some models are easy to address these questions. For example, always the same, but the model species such as C. elegans, you can, using CRISPR-Cas9, you can insert some, you can modify genes, and you can also insert epigenetic modifiers inside the genome. So if you want really to address an epigenetic DNA modification as a promoter of a gene, it's something that is possible to do. So to study specific gene methylation, well, I don't know if we are at the level that we can do that, but if we have to think of the future, it's something that could be also used. And yes, and then yes, the chicken model, it's also a
Yeah.
good idea because it's something that may be shared also with the environment, because we don't have so many data on birds. So
Yeah, I have the review paper from them, but I know the group that does it. I will I will put it. Allez, where is it? How can I have the paper on its own? Can I share it in the chat, or I will put the where is the chat?
About question in the meantime when you are writing the reference about question number 14, how many generations should be followed? So this morning we have discussed about that also. And for environmental species, it always depends on the reproduction strategy, because organisms that have an external reproduction, if you go from generation F2 after F0 irradiation, you are addressing cells that have never been irradiated, and for the other organisms, it's from generation F3. So this morning we also discussed about definitions of transgenerational and multi-generational exposure, and so transgenerational, we agreed on a definition that would be it's an exposure where offspring itself has not been exposed either at the somatic or germ cells. And for multi-generational exposure, it was a chronic or acute pattern exposure that comes back after several generations, continuously or not, because you may expose the organism, the whole generation from birth to death, or only some important life stages, but it's still multi-generational because different generations have been exposed. And for human, it may be the case if you have a medical exposure of the parents and then of the children and grandchildren. Or
if I could just add to that, Christelle,
yes.
During our definitions, one thing we wanted to clarify was that the main difference between trans versus multi-generational is that, as Christelle said, for multi-generational, oftentimes it's a protracted exposure. It doesn't have to be, but what we decided on was it's the same pattern of exposure. So whether it's acute, fractionated, protracted, across multiple generations. And the key we kind of decided on was that without missing a generation. So if you're looking at F3, all the generations leading up to that F3 were exposed. Whereas transgenerational, we said kind of the distinguishing feature is that it's an exposure of a previous generation and that no cell in the organism was exposed at all. So for example, if it's a pregnant mammal, the germ cells are there if the animal's pregnant. So that's sort of our distinguishing feature, is that no cell of the organism you're observing was irradiated directly for transgenerational.
Okay, then about the question 11, transferability of results from animal experiments to humans. So that's a big question actually. So we have learned that the mutation rates in the specific locus tests are higher than the mutation rate from the recent whole genome sequencing studies in humans. A simple question actually. Would it be simply possible to use a correction factor for this?
Who wants to answer this simple question?
A very simple question with a very difficult answer. I really don't know.
If I
It
could
would
add
be nice. Cheyenne, go ahead.
my one thought for this is like it is kind of an impossible question to answer. I would argue where I think we can control things is study design. If we analyze a specific study, whatever the model system, if that specific study was able to control variables correctly in an appropriate way, and the exposure of radiation is the intervention you're directly testing, I would argue it's relevant to some degree to humans, right? It may not be fully relevant, but to some degree it's relevant. So if we can't pick a specific answer, maybe we should focus on what criteria are we looking for? Maybe within studies, are there specific features that we should be analyzing? Just my thoughts about this.
Yeah, if I may add, I agree with Cheyenne, and I think it's not possible to transfer risk factors. That will not be possible. But you can of course under controlled experimental conditions you can test the impact on confounders, and you can also look at the shape of the dose response, which I think is quite relevant.
Yeah, Cisco, for the correction factor probably can be used for the doses, because for each organism, the dose, low dose, high dose are totally different for human situations. For example, C. elegans probably needs, kilograms of radiation exposure, right? Same is true for crustaceans, for example. And then mice, we have different definitions for low doses in each organism. So for that, probably we can use a correction factor. What human dose relevant for the other systems?
And just to add to that, one thing that we discussed earlier today was that a big thing is it really depends on what you're analyzing. If you're asking a fundamental mechanism question, whether it's kilogray in a C. elegans versus milligray to gray in a human, if fundamentally you're asking questions about something as simple as DNA repair, then I think it's appropriate. But I agree that it's very difficult and I think kind of incorrect to just apply thresholds, that level of translatability. I think you have to be careful in what you translate, but I think if you're upfront and focus on what you're comparing, that's, I feel like, the best you could really do.
Agree.
And also, it's something that we discussed this morning about if we want to study mechanisms and the way it can have a consequence in terms of adverse outcome. It's also something that can be used in the concept of AOP, where you may identify important molecular or cellular mechanism that can ultimately reach adverse outcomes or pathologies. And the interest also would be to not really to transfer the animal results, but to compare the different responses in different organisms to have a stronger confidence in the main mechanisms. If it's if you find the same mechanism, the same critical mechanisms in different organisms, I think it makes yes, for example, yeah. Mm. Mm. Mm.
Perhaps, well, I can ask questions. I have more questions. So, thinking about the adverse outcome pathway and epigenetic effects, what would be the initiating event?
I think still the initiating event, even for the epigenetic effects, can be the interaction with the DNA. So like what we the deposition of energy, as we have within the AOP working group on reproduction, we have put deposition of energy as the initiating event. That leads to DNA damage, that can lead to change in methylation marks, that can lead to ROS production, and so on. But that was actually Within that working group, if I'm not, but please correct me, Sandrine, if I was wrong, that was the consensus where we came up with.
Okay. And Nelo, it was a lot of discussion in this group to identify.
It was a lot of discussion. It was a lot of discussion
to find out to agree to a same molecular initiating events.
Yeah, yeah. But that was the one that we finally
we agreed
on. Yeah. But it's true. It
I'm
was
not sure I understood correctly. So the event would still be energy deposition in DNA.
No, energy
No,
deposition.
okay, in general.
Just energy deposition in matter in
So,
your
so
organism.
it doesn't specify in which molecule.
No.
Does it have to be your subject of interest? If you're looking at a cell, if you're looking at an animal, does that specific cell itself have to be irradiated? Or sorry, does it have to absorb energy?
No, I think you can have if you point towards bystander effects, you can have that if there's somewhere a link. Yes, you don't have to have the irradiation in itself in that cell. I think.
Can I make a comment? I think I'm not sure if I would buy this answer, because what you actually say is that it's a radiation effect. Yeah, well, yes, it's a radiation effect, but just putting it in a different way, saying energy deposition. I don't think we should say this, because this is, well, it means we have really no idea. And then let's say we have no idea, but maybe because this could be membrane effects, changes of in responsiveness of what is sitting on the membranes, receptor proteins and stuff like this. Maybe oxidative stress. Maybe effects on the mitochondria. Perhaps we should list them. Possible effects.
Yeah, but in the AOP you list them, but not as an initiating event, but as a key event that follows the initiating event.
Okay. Okay. All right. Okay. Right.
Yeah, I agree that deposition of energy in itself may not be very informative, because yes, it is a bit of a circular reasoning.
I would argue that, like I get what you're saying, but you need that energy deposition to initiate something, right? If your system is sitting there and no excess energy is absorbed, in theory, nothing should happen.
And I think even for DNA damages, it's not so clear for me about the role of where is the important energy deposition. We know it's on the nucleus, but what about the role of mitochondria? There are papers now coming out saying that there can be an impact. And I think that for epigenetics, it's a mess. We don't know anything. We don't even know if epigenetic, we were talking about that at lunch, but if DNA methylation is really the cause or the consequence of all the response, because it may be a different interaction of ionizing radiation with which triggers DNA methylation, which then triggers gene expression, etc. So it's we are far from knowing that, and perhaps the only way to do that would be to find a way to reverse the mechanism, to prove that it's really by blocking DNA methylation on this gene that we don't have the adverse outcome. But it's a big job.
Yeah, if you can work like you said this morning, that there are for C. elegans, that there are mutant strains around, that if you can work with a mutant and you see that you don't have the adverse outcome when there is a pathway that is blocked, then yeah, that is a way to prove it. But for Arabidopsis, for example, we often use mutants in a to prove that it is an essential step to go towards a mechanism or an endpoint.
So how to study? Could you use microbeam experiments to study the targets?
Yes, yes, it's true that we discuss also about using microbeams either to target subcellular components, mitochondria, membrane, nucleus, but also to target different maturation germ cells with different maturation stages, and then it's possible to really study the sensitivity of each mature stem cells. And I don't know, because I'm thinking back about the discussion with Professor Nakamura, but I don't know if it would be possible locally to induce an hypoxia on a different tissue. I don't know. I'm thinking loud, but stop me.
Well, just a
Well,
comment.
I'm specifically talking about the oocytes, not ordinary organs. Just comment.
But the hypoxia in oocytes, it is supposed, it will be chronic, right?
Yes.
Yeah, and that is very difficult to simulate under in vitro conditions. To study
Oh,
this.
not really so. I think it's feasible.
Yeah, but there we expect. I would expect to have different responses following acute and chronic hypoxia, because chronic hypoxia doesn't.
Acute hypoxia is often discussed among oncologists, right?
Yes. Yes.
Because blood pressure goes sometimes stop. But in the case of immature oocytes, they are chronic hypoxia. But why? Anyhow, to make a discussion short, we know basic mechanisms why immature oocytes can stay there for, in the case of humans, almost 50 years, staying there alive. And I'm guessing the mechanism, metabolic, mechanism may be quite different. They are not suffering from lack of oxygen. Those immature oocytes are staying there happily. Low level oxygen is probably comfortable for them.
Yes, but it means they are deployed of energy, and that means they may actually be more radio sensitive. This may sensitize them then rather than make them more resistant. I mean, the low level of oxygen, chronic hypoxia makes cells actually more sensitive to radiation because they don't have enough energy
No, I don't think so.
Okay, that's it. We can carry it out among ourselves one time.
Yep. Or maybe after drafting, then we may discuss on the draft.
I know, but the mechanism should be the same because your cells have no oxygen. They are.
Yeah, I'm just highlighting one preprint I mentioned yesterday about the uh, epigenetic factors, right? It's just, I think, submitted by John Schumacher and his team, DNA repair thing, and there they find transgenerational effects and reversal of transgeneration effects when they use mutant C. elegans, mutant for H1.0 ortholog HIS24 and HPL1 mutant. So in those transgenic worms, the reversal effect was seen, and it's not published yet, but in the preprint server. Yeah. So they. Yeah. Yeah, but the only problem I have with this paper is they don't mention the dose. I didn't see in the material methods. Probably it's in supplementary. They just use ionizing radiation, just like any DNA repair. And they claim that there is homologous recombination repair.
So apologies for being ignorant with the current state of understanding from animal models, but there are studies that are showing what models are showing effects in the literature, and what is it about those models that we think that we need to look for other models?
About the transgenerational effects and epigenetics or in general. Yeah. So for the moment, most of results that have shown transgenerational effects with ionizing radiation have been obtained with invertebrate species. So the microcrustacea we were talking about, and nematode, C. elegans. To my knowledge, in fish, real transgenerational effects have not been already shown. On sex ratio, more males appear, and it's something that we also have in using invertebrates. And for fish, what we have seen, it's epigenetic marks that have been transmitted, but they did not came with an effect on fertility or abnormality in offspring. So to what extent can we say that these molecular changes are really significant at the level of the individual or an adverse outcome? It's still not clear for me. Whereas for invertebrates, we have evidences of transgenerational effects in terms of reproduction and also molecular mechanisms.
So the mouse model, the mouse experiments, are they all higher doses as well, the ones that have been done? So I guess I'm just trying to get a sense of what has been done in mouse models, and well, I guess I'm trying to stick to more mammalian. And what are
Yes, it is known that quite a number of factors can have epigenetic effects or transgenerational effects even. So realizing that there is lack of knowledge on radiation effects, what can we conclude from transgenerational inheritance in general?
There is the study that was presented by Zenko yesterday on the effect of visocycline on I think it's 4 generation of mice, and so they have I think much more than 4, because this paper, it was performed with an endocrine disruptor, and it's Skinner et al. And they have shown transgenerational effects in mice, but again, they don't have any explanation of the mechanisms. So it may be we could say it may be a correlation, or I don't know, but and for ionizing radiation, in the field there are data showing that, but rodents have been exposed for several generations. It's not real transgenerational effects.
Yeah, I think Yoshi, in your institute, there are some multi-generation, transgenerational effects are being studied in mice, right? Is there or no?
Yes, yes.
I mean, are the papers published now or are still in preparation?
Yes, some of data have been published in last year. Yes, and our experimental protocol is chronic exposures, 20 milligray per day for 400 days, and the total dose is 8,000 milligray, 8 gray. And under these situations, the genomic deletions as detected by RACGH, the frequency of deletion was indeed increased by around twofold. And also in parallel, the offspring have a little bit shorter lifespan. But in case of the low dose rate at 1 milligray per day, or milligray in total, have failed to induce the deletion nor lifespan shortening. Of course, 0.05 milligray per day, 20 milligray in total, have negligible, almost no effect for genomic deletion nor lifespan shortening. Yes. Is it okay?
Yeah, sure.
Have you answered your question?
Yeah. I was wondering whether the study has been completed or the paper has been published. And you mentioned that only couple of endpoints are studied, or you have a multi-parametric study.
Just lifespan shortening and cancer induction and the DNA deletions in offspring. Yes, these three parameters.
Okay, thank you.
Also, I have a question because so the interest of coming back to the best biological model, one of the interest of using other animal models is that you can directly analyze ovocyte genome and sperm genome. Of course, it's impossible in human. So I don't know how many studies have been done really analyzing the transfer of DNA damages or other factors from the gametes. Because generally, studies analyze, for example, DNA deletions in parents and then DNA deletions in embryos or offspring. But is there a causality link by analyzing, is this causality link is made by analyzing directly the germ genome, germ cells genome? Is it done? Or?
No, we haven't done yet.
Yeah. Probably for chemical exposures, I think people have done such experiments directly on sperm DNA, but I don't recall on using ionizing radiation, but probably that's something to think about. Right. What about zebrafish? Enough data available for?
Now it should be possible, but the problem before was always to have enough biological material to analyze, because zebrafish, it's 1 centimeter length. you can get some sperm by using specific tools, etc. But then what is done usually is the study of sperm motility or sperm morphological abnormalities. But to analyze DNA directly from sperm, now you can have single cell analysis, and with tool like that, it would be possible, I think. But even from only sperm DNA would not be sufficient, because you can have a non-coding RNA that is also transferred from germ cells to the embryo. So all of that factors contribute to the transgenerational effects. But it's perhaps a direction we have to take. Huh?
Yeah, I know that in Osaka, Todo was conducting a massive experiment on zebrafish multigen. I don't know what happened to the. I visited his lab few years ago, and he has a big, lab with yeah, huge numbers of zebrafish model. I don't know whether they have published any data on transgenerational effects. Yeah.
I'm just wondering about the sampling of the human sperm, for instance, work that's in fertility clinics, whether there's any sort of, so there's quite a lot of papers on DNA damage and looking in sperm directly for fertility work. And I just wonder if that's an avenue for us to explore. For instance, if individuals that have different exposures or therapy, and then there's some sort of link up with getting samples and understanding from that perspective, just as a possible place for human sample.
Well, that might be an option, and of course, if you what has been discussed in many countries recently is that the sperm quality is going down, and what might be the reasons, and different also epigenetic mechanisms have been discussed, not so much genotoxicity.
Well, yeah, but basically the literature that would be surrounding fertility, the literature, but also what fertility clinics gather in terms of information and store in terms of samples.
Okay, so I think that we should finish this discussion by the coffee break. So should we go to the question 12? Which endpoints and or surrogate markers are likely to be the most informative? Experimental models as surrogate for, I don't know, human risk assessment. Of course, there are the genetic endpoints, Mendelian diseases, chromosomal aberrations, chromosomal syndromes, congenital malformations, and multifactorial diseases. What do you think? And do we have experimental models to support this?
We talked about this morning, and in fact, even studying reproduction endpoint, there are a lot of endpoints that can be chosen, but they are diverse among the different species. Yeah, fertility, fecundity, hatching rate. Well, it's different for different models, but What is not easy, perhaps Professor Nakamura, if you want to answer.
Just a question. Well, I'm a little bit confused in that markers. Well, well, I can't help feeling that genomic change can well be a markers for the genetic effects. I mean, because it's simply an alteration of the genome does not always lead to diseases. But this question is a little bit difficult for me. Well, well, markers means like a blood pressure for the risk for heart disease, something like that. But markers for genetic effects of radiation, what does it mean?
Yes, I think this is we can first of all think about that, if this is a relevant approach at all.
Mhm.
And that if we think about preconceptional exposure, should we think about also something else than genetic effects like fertility, like is done at the Biota group? Reproduction is there important, but I don't know. Surrogate markers, if you cannot directly measure something, but you would have something that is informative.
If I could add my thoughts, again, all of these questions are impossible to answer, but I think one thing you can do is you can use non-human models to guide your questions, right? Because something like, say, transcriptomics, you're going to get a wealth of data. What does it actually mean? Maybe you can say, okay, in this fish species, they identified specific biomarkers, again, within the context of fish, but maybe if all of them are related to important genes in humans, it gives us justification to do detailed screening for those genes. Just a thought, because we talked about epigenetics is not new, right? This has been in nature since for forever, but we've just more recently started to focus in on it and give a lot of attention to it. But I think it really depends on what you're interested in. For the animal side of things, we talked a lot about, populations, right? We don't generally concern ourselves too much about very specific, subtle changes in a specific individual. What we care is, could these markers indicate, population dynamics, stability of populations? If you're trying to look for human health outcomes, I think the best thing you can do is find markers that are easy to measure, that are sensitive enough, specific enough, all of these parameters, which are almost impossible. And I think the best thing you can do is use these approaches to guide your investigations. That's just my general kind of thought for this. And I also think it's important to ask, what type of exposure are you worried about? If you're worried about a very huge, acute, kind of disaster kind of scenario exposure, the outcomes of interest would probably be different than someone who lives in a high natural background area, right? So the exact nature of the exposure matters, because then it will dictate what types of effects you'll want to investigate as well.
No, I think we discussed this yesterday, right? So all gene expression data and how they interpret, even they don't even segregate them into different pathways. There is gene ontology, pathway analysis, but that ultimately in the conclusion they don't even mention whether this is going to affect the human well-being in the long run, and how long these genes are upregulated or downregulated, right? So most of the gene expression studies are done for biodosimetry work. It's not for disease prediction or other things. So and most of the expression changes are again, as highlighted here, it may depend on dose rate, dose used, and time post exposure. Because yeah, and then cell type. So it can be even transient. H2AX is one good example in that sense. It's upregulated immediately after exposure, and within 24 to 48 hours comes to basal level, right? So you can't even predict anything using that expression data, but who knows? If you do a long-term study, probably then you will have one set of genes are upregulated in the beginning, and then after 7 days or 14 days, you may have other set of genes which are down or upregulated. Probably then you can relate that to a particular what's the term they use, AOP, right? Adverse outcome pathways, whether these genes belong to those pathways which are ultimately of functional relevance, right?
So I agree that what are the endpoints that are likely to be the most informative? You need to have endpoints that have that are reliable, that can be linked to an effect at the function or macroscopic effect, something really strong on well-being. But on the other hand, the studies that we have performed on the zebrafish embryonic development, we have studied the development of zebrafish over different dose rates, and we have not seen very severe effects on abnormalities, for example, very subtle effects on development rates or hatching. But on the other hand, we have studied transcriptomics and epigenetics, and both so transcriptomics and epigenetic pointed to the same gene responses that were linked to retinoic acid, oxytocin, so different proteins that are important for neurodevelopment or neurotransmission. And then after that, we came back and we did behavioral studies. And using those studies, we saw subtle effects that were not seen at the level of the whole development. So I don't know, perhaps we are learning while walking, but sometimes you have also to adapt your endpoints to the level of sensitivity you want to tackle. Because if you are addressing a very accidental release of radionuclides, it's easy. You take survival, you take reproduction, you will always see something. But if the question is to see effects for chronic low dose exposure, then perhaps you have to think more about what are the new endpoints that you have to follow in order to, perhaps it's not always DNA repair, perhaps it's cardiovascular disease. You know that much better than me, but cardiovascular disease, neurodevelopment, neurodegeneration, I don't know, but
okay, so kind of summary, it would be endpoints that are relevant for the health outcome that you're interested in. And at high doses, it's easy because there are things to be measured and there are effects. But for low doses, this is much more difficult.
And the studies and data from high background areas are not conclusive enough to prove even transgenerational effects, right?
I think just kind of thinking about transgenerational models, one thing that makes it relevant is, like we talked about, if it's a very subtle genomic level change, the common question is, well, what does it actually mean functionally to the offspring? But one interesting thing about transgenerational effects is that it shows you long-term concerns, right? We're talking about exposing a parent, and then multiple generations later, even after you stop the exposure, you're observing an effect. From a human protection standpoint, that's concerning, right? Because now we're talking about, okay, if you go get the CT scan, sure, you won't get microcephaly or something that requires a high dose threshold. But are you concerned about your great grandchildren 50 to 100 years from now potentially being affected, right? So I think that's why there's interest in transgenerational models. But again, to tease apart and translate a lot of these models to humans is the challenge. But I think it highlights an important aspect of radiation protection, at least, in that it's a long-term concern, right? You're looking at generations, literally entire life cycles of an organism, not within a single lifespan.
So in the summary, we just said that high dose was probably easier than low dose, but if it is about pre-conceptional exposure, I think even at high doses, it's difficult. So if it's exposure during pregnancy, yes, then high doses are easier, but pre-conceptional, not even high dose is easy. Sorry?
No, the thing is, if the embryo survives, that means that's fine, right? Even if even the radiation effects is transmitted for multiple generations, that means that is not detrimental because they are fertile, whether it's the animal model or human.
Yes, well, severe dominant mutations. Yeah.
Detrimental. I mean, mutation is for survival.
Right.
Evolutionary point of view.
Yeah, and recessive mutants may persist, but if you have it heterozygous, it doesn't matter, mostly. Okay, so to move on, question 13. What are the experimental strategies to investigate the dose and time dependence in progression of germ cell damage?
So could I clarify, when you mean time, do you mean time post-exposure or the developmental period in which you're irradiating?
It can be both, right? Yeah.
So one thing we talked about earlier was we kind of concluded there's three important variables that you always have to consider, or generally want to consider: dose rate, and time of exposure. Because the way you look at it, one gray, depending on where you are in development, can do very different things. So that's what makes this complicated. You have to consider where that organism is in its life cycle, because that affects its sensitivity as well. So I think.
So it's the development of the spermatogonium, is it? So how in humans, however many weeks or months, and then thinking about the experimental model so that you're actually assessing the same stage of the sperm cell development.
I would argue that's where rodent models let you answer that question, because you're able to experimentally time when they get pregnant. You're able to control those variables. Again, it's not a perfect translation, but I think you can get some information out of that, I would argue.
Yeah, that brings back to the question Professor Nakamura asked yesterday. Nakamura. Yeah, why we see transgenerational effects in animal models, and that's not reflected in human studies, right? Andre, I answered that yesterday or today in the discussion, that most of the animal experiments are well controlled. You don't have the other variables. Even mice, maintained under specific pathogen-free conditions. You don't have any other exposures to even, conditions which are adverse to growth, for example. But humans, we can't do that type of thing because they are open in the environment, right? So probably that's why we may not be able to see the effects in humans.
So do you think field studies kind of give you some information about that? Because now you're in the field, you're exposed to all those other stressors in the system. Could we make the argument that they give you some information?
I don't know. Probably the Chernobyl biota study probably may. No. It's not my area of expertise. Anyone has done wild animals.
But if you do experiments while animals, yeah, but then it's chronic exposure. It's not transgenerational. So I think we can forget this. This is not the question that we are interested. But I think one difference or one additional aspect to what you have said, Prakash, about controlling the conditions is that with mice, we are working with inbred strains that are genetically, well, kind of uniform, and then we can potentially, or we select strains that show a particular susceptibility or susceptibility to particular endpoints, which are much easier to study than in humans. And I think that's, so we kind of artificially potentiate the effect in order to see it, which is okay. I think, well, it's the way to go, but that's why we can't use the results to transfer risk.
It's also the lifespan of the mice and the controls. You can control much better what kind of controls to use, and you can validate your results much quicker, which is not possible in humans. So these are all arguments.
But at the current level of knowledge, it's the time dependence in progression of germ cell damage, not necessarily in humans. Would it not still be good to then look at those animals so at least we understand what happens there, and we can then perhaps use those experiments to formulate a hypothesis on what happens in humans? So still to go with the animal models and then see the humans as a next step?
That's definitely one thing, that mechanisms can be only studied in, I mean, well, not only, but mostly studied in animal system.
I think I could also flip your argument and argue back. If the question is fundamentally, what is the effect of ionizing radiation? You kind of have to do it in a lab, because then you can control all those other factors. Because in a more natural setting, you're right, there's all these other variables. You see a response, right? You see an effect, you statistically test it, you see an effect. You then have to go back and argue to me, say, I'm reviewing your paper, and conclusively prove that it was the radiation, right? And that's always tricky to do. So I think that's where lab studies are really the advantage, because you're very directly asking about the effect of really only the radiation in theory. So I think there is value. Again, you have to be careful of how to translate it, but I think there is value in all these animal studies, because the other thing is, the more people do them, the more statistical power you start to see, because you have more sample sizes. Maybe you can draw some conclusions within a specific model.
Yes, well, thinking about the mouse experiments and the mutation frequencies that are higher than those in humans. So one reason is simply that there were loci measured that had high frequencies of mutations, so sensitive loci. We easily study things that where we see effects, and that's in a way sensitive strategy to study mutagenesis in mice. But transferability then to radiation protection is another issue. But there are also whole genome analysis of mutations done in mice, and that's the same conclusion as in humans, to my understanding. So mutation rate is much lower than those estimated from the specific locus tests. So in that way, going to the same approach in mice that we are using in humans now in the TRIO studies might help the transferability. Go. That was, well, still question 13. Well, yeah. Dose and time dependence.
In vitro or?
I should know, but I don't remember what we thought about that.
Sorry. Well, but it also could be sampling time.
Let's go to the next one. I think we have to do this before coffee break. So how many generations? That was already discussed, I think, yeah. And which organs should be studied?
I don't understand this question. What do you want? What I mean, obviously we want to study all organs, or anything where we get a response.
Yeah, I would argue any organ. If for humans very specifically, we care. I think we care about every organ.
Yeah.
Yeah, because that was easy. I mean, it's anyway. No, it's whole embryo exposure, whether it's preconceptional or in utero exposure, right? Yeah, F1, right? Which organs in F1? Yeah. Yeah, exactly. So entire. Yeah.
Are you talking about the parents or the offspring? Affecting the parents or the offspring? We don't.
Well, we can split that. Well, if we want to study the parents of the burn or size, yeah. But then in the offspring.
Yes, and yeah, all organs.
Question 13 was the parents. And then
Offspring. Three. Is that your consensus in your group? Yeah. Okay. Yeah. Yeah, sorry.
I'm sorry. Okay, no, yes, I was thinking once we are looking at the population level, we can go with Baota with 30 generations, and we can see an effect after 30 generations. But it's a very different output that in human.
In animal experiments, when you say that you're studying multiple generations, so you sacrifice the animals to look at the mutations, or you are using samples, blood samples from the living F1, F2, or F3, because you need to sacrifice. So then you are. But you expect the radiation effects in all F1. There is no selection. So you expect the same thing to happen in the surviving offsprings.
We follow, it's perhaps the question is different than for humans, but we follow the adaptation. So the animals that survive at each generation are the stronger, and we don't select them. It's a dilution of the population. It's a Darwinian selection. It's a dilution of the population with the stronger or the faster reproducing animals. And for nematodes, what we have seen, in fact, is that males appear to be dominant in the population because there are several reasons, but because there is it's complicated, but nematodes are hermaphrodites, and so they can self-fertilize also. So when they do that, they are not limited by the sperm numbers, and well, there are several evolutionary reasons, but well, perhaps it's out of this discussion.
The last question.
Yes, the last and important for the TG 121. Compared to the state of knowledge in 2007, what is the contribution of new results, especially from epigenetics, and their potential impact on risk calculation for heritable effects? 0. For heritable effects. Oh, risk calculation, epigenetics.
I don't think so. But I think this is the question why we are here. So I think we can only say, well, we will answer this at the end, or TG1. Yeah, that will be the outcome of the work of the task group.
How long does a task group? When's it? 3-4 years.
Yeah, so I don't think we can do anything about calculations, but we can say where progress has been made that might affect these calculations. So is there more information now that should be taken into account when doing the calculation? And because I don't think we can come up with a risk calculation here now, but if someone else does it, what should they take into account, and what is new since 2007? Yeah. Yeah. Yeah,
Yeah,
or and their potential impact could become that could potentially have impact on. So that we don't quantify it, we just specify.
But are we talking about experimental models, which means these are not epi studies?
I actually think that the whole genome experiments are quite important. There is at least one study on mice measuring the directly the mutation rate after radiation exposure. Of course, we can say that the analysis should be. There were some comments on the detection efficiency for deletions, so that might be a question mark. So it's.
Yeah, I think it's also, in the last 15 years, because if you look at 2007 and 2022, it's 15 years. There's a lot of knowledge gathered from a lot of lab studies, but also from field data that shows that epigenetic plays a role. But
Yes.
how to include that at this moment in risk calculation, that is something that is very difficult to say at this moment.
And if it does really have impact on the risk, because the health outcome is probably the same, but what is caused by genetic and what is caused by the epigenetic mechanisms, that's the question.
Yeah.
But it is a new mechanism that has to be considered anyway.
Yeah,
So
and that was the focus of research from the last 15 years for many.
And since these 15 years, we have new tools now and new result from genome-wide sequencing. It was not used in 2007, and we have to take all the information from these new tools.
New tools, and then perhaps using the same tools in animals and humans for the comparability. That might be the advice. Yeah, I think that's a good recommendation for before coffee.
In Belgium, there is coffee.
We're on our way. There's a bar upstairs.
Yeah. Yeah, so we are going for a coffee break, so we'll be back in 20 minutes. 10.
Christelle, do we resume in our small group or? Christelle?
Yeah, the last part is mechanisms. Yeah, yeah. Yeah. Yeah, and I think we need to at least start thinking about the structure of. Yeah. Okay. Thank you for joining this session, and I think we all had a good discussion, right?
I think it would be useful to split because we have question for the relevance to population levels, and it's really specific for us. So we meet in our last room
Zoom.
in the C room.
Yeah.
Thank you.
Okay.
Yeah. Question 20 is.
Yeah, compared to the state of knowledge, I think this we try to answer from the different perspectives of epidemiology, experimental models, mechanistic approaches. That's kind of redundant, but there are also other. I was just thinking if we can pool together those questions that are kind of redundant in the different sections, so that answering same question from the different perspectives. Epidemiology, is that possible? Not. Yes, exactly. Yes. Yeah. 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 Good plan. So.
Maybe later. Maybe
Yeah,
later when the
yeah,
talk is ready.
yeah. Let's finish this. Just trying to compare the questions here. But anyway, let's go question by question now and then try to pool them. Question 17. Which are the driving mechanisms that should be explored to improve understanding of radiation-induced effects on germ cells and next generations?
Yeah, so also I think one thing that we could envisage is genomic stability, because it may be that by exposure of germ cells, then we induce some changes that lead to genomic instability. And how can you study this? Here I come back to the idea of irradiating cells, taking cell samples, irradiating them, and then under in vitro conditions and studying their response. One dose looking at DNA damage response.
Well, that's an obvious mechanism, I guess. Yes, please.
I agree that we can take the cells and ex vivo or in vitro do these additional exposures. Another way to look at the mechanisms is that when we analyze whole genome sequencing results, we can actually have an idea when we see some indels, some structural rearrangements, we can have an idea which DNA repair mechanisms or deficiency in which DNA repair mechanisms are responsible for specific mutations. This is what has been done in the Chernobyl cohort thyroid cancer study last year, which came at the same time with the transgenerational effects. I think that's an exemplary, it's an example how to do this kind of analysis, because they concluded that it's non-homologous end joining which is responsible for those rearrangements. So that would be useful. And just a general kind of comment question. I'm understanding that now, before we've discussed what to look at, what to pay attention to when we analyze the literature from 2007 to draw some conclusions. And now we're moving more towards like what to do in new studies or what to recommend for new studies. Is that right?
I think if we're writing a review paper after this, I think we should also look back and then probably with 2007 as a starting point. So look, yeah. Oh, true, yeah. Yeah. But yeah, there should be a review aspect as well. Yeah. Yeah, so that would be, yeah. Yeah.
But my understanding, it's a bit of a combination. I think all of these questions are a combination of both. You know, what have we learned since 2007, and also from that thinking about where to go. So I think those two things are implicit in most of the questions.
At the same time, many of the questions that we explored so far, we cannot answer explicitly now before actually completing this review by the task group for future recommendations. So I think it's just the information is incomplete to address many of the previous questions. It just
when we start working on this structure, then probably to keep this question in mind, you know, ultimately that has to be mentioned somewhere, and the future suggestions, for example.
If we have studies that are not concordant with each other about in relation to the effects in next generations, how we can think about mechanisms, because some studies showed and the others did not. So it may be more straightforward for the radiation-induced effect on germ cells, but radiation-induced effects in next generation, I think how we will decide we trust this study and then we think about the mechanism, or I'm a bit, you know, puzzled.
I would address this question. I mean, given this is a review, you also somehow summarize what has been done, what kind of mechanisms have been used so far to explore. And from that, you sort of becomes, or hope to become clear what mechanism needs to be explored in the future, which were not conclusive, which needs to be completed, or so shortly, this question has to be answered based on what has been done before and what are the gaps that before were not identified.
I think also these questions, they're a tool. They're intended to help us structure the discussion. So if we think it's not quite the right question, we can deviate from it. It's not that we have to answer everything on the list. Yeah. And so we can also conclude that we don't know it. Yeah. So we could also say, well, to answer this question, we need to summarize the evidence, identify the contradictions, and then formulate something. Perhaps we don't know it now.
Since we are speaking about the mechanisms and knowledge gaps and future studies, I also have another general comment. I think maybe this discussion also struggling a little bit, the after lunch discussion, with the lack of focus, which is a common feature of radiation protection research. And what I mean is that very often research projects do not formulate properly hypotheses or specific questions, because we discussed endpoints, which endpoints, how many generations, and other questions. I think future research should try to research projects should try to formulate better hypotheses or specific questions and then address them in a very focused and specific way. And it should be okay that it's a simple question, simple hypothesis. But if you ask too generic question, like an example of this generic question is there a transgenerational effect? So what is an effect? So, but if I say, is exposure of parents inducing neurological disease in F1 and F2? So I can test that in the experiment. But if I'm asking a question, is there a transgenerational effect? Then I can measure one endpoint. I can measure expression of one single gene. And if I find a difference, I say, yes, there is a transgenerational effect. Okay, so I think we have to be careful how we formulate the hypothesis or specific questions. And then step by step, study by study, we will be building this knowledge base to answer the question, are there transgenerational effects? And if there are, what kind of transgenerational effects? So this is a generic question, which is important for future studies, I think.
Yeah, I think most of the studies are observational in nature. I suspect there was any study focused on understanding the mechanism, how even Dimitri, your point on neurological effects, right? The reports say that there is a transgenerational effect on neural development, for example. And I don't know whether anyone has studied what is the mechanism to produce such effects, right? Yeah. 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 I mean, exactly. Yeah. Yeah. But when we, I mean, mostly in radiation biology, it is observational studies where we want to look at the effects and the data. Hypothesis is formed afterwards. Yeah, I mean.
So, yeah, but that happens.
That's how it is. Yeah.
Yeah.
But people who study the mechanisms, they always go with the hypothesis and then come with. Yeah.
Yeah, you can disprove your hypothesis. It's okay. Or you can formulate a new hypothesis at the end. It's okay too. But I think you should be starting upfront with the hypothesis. Or sometimes hypothesis is not possible. You can ask a specific question. Yeah.
Yeah,
For example, is genomic instability involved in neurological disease?
I think that much of the mechanisms that we are actually discussing do not necessarily come from radiation studies. So then you can build on this general knowledge and make the hypothesis for radiation. So if this operates for after radiation exposure as well.
I fully agree with what you said. I was just wondering what poor Fika has to write down. So maybe we can make the recommendation that future studies must be more strictly hypothesis driven, something like this. Yeah, because that will be then for tomorrow for the reporting.
Yes, yes. Yeah, in fact, there is a big world outside the radiation field. So radiation discipline is quite narrow in the end. So we should make use of the science elsewhere. Well, what add here? Well, we can probably come back to it after we have gone through the other questions. There is a question on mathematical models. What would be useful? How might they be best integrated into a mechanistic framework? Any ideas on this?
I think a very obvious place for mathematical models would be in linking results in animal models to humans. So you were saying, well, can we just have a simple factor to go from one to another? No, I don't think we can do that. But I think mathematical models probably could help there by identifying what the systems have in common and where the differences are, and possibly also identifying where the knowledge gaps are, and we would need additional experiments. So I primarily see a role for mathematical models in linking those two, and in possibly also in getting from the experimental results to risk estimates. Because there will be a lot of epidemiology in there, but mathematical models could complement those.
Okay, anything more here?
So in mathematical models, we immediately talk about the dose response, which is one of the basic questions, the shape of the dose response. So I think it's important for future studies if you can use multiple doses, spanning the whole region of interest. That would be one of the recommendations. If you're designing a new study, try to do dose response. Yes, dosimetry.
When you have all the, the dose range and you plot your observations and just try to describe it using whatever linear, log linear quadratic threshold. It can be all kind of models that will help to establish the association between the gonadal dose and the whatever endpoints we looking at.
Well, wonder about population genetics. That might be informative, but I'm afraid we are not very good experts. What, Rona? Any view on this? We are still thinking about the mathematical models.
Yeah, I think the dose responsing is probably one aspect which is important. So we have been discussing about epigenetic mechanisms, right? Whether changes in epigenetic pathways follow any dose response. I don't, I mean, yeah. Because other things we know, right? Genetic changes, genomic instability, for example, chromosomal changes, DNA damage. I think we know pretty much about the dose response. Yeah. 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 No, same. Yeah. Mm. similar to transcriptomics data because at different doses, different set of genes are upregulated or downregulated. Yeah.
I think that could also be a recommendation just in general to start with a simpler setting. And then probably mathematical models can help to formulate hypotheses to guide experiments. And I do see a role for them there. And of course, there's also possibly data analysis, because there might be a lot of data available. And it might be possible to ask smarter questions based on your data sets with mathematical models. But that's not my personal expertise, but I would expect that.
Yes, I agree. Mathematical models are important. Algorithms of analysis of the data. If you have 3 million CpG methylation profile, you need mathematical models how to interpret that. And I remembered in this regard, I remembered this methylation age clock. It's, yes, biological age based on the methylation profile. There's some error associated with that, but I think it's pretty much established in aging research in gerontology that it's a good predictor of biological age. So we can probably borrow some of the concepts from aging research and try to apply to radiation studies on transgenerational effects and see if there's any correlation of this existing. This is a good endpoint. Just take it, borrow, and see if it's changing.
For the analysis of big data, the artificial intelligence and all these neural network type of approaches are quite popular. So maybe these could be considered. That might be one way of contributing to the next call, actually, because they are considering the artificial intelligence. But I think it's mostly in areas like dosimetry and nuclear safety and that kind of things. But there are applications in biology as well. Okay, so that might be worth for a workshop maybe to look into that and invite some bioinformatics experts. Shall we go to the next one? So what is the relative contribution of genetic versus epigenetic mechanisms in disease development? And these are the classes of hereditary diseases that has been used in the past. Mendelian diseases, chromosomal disorders, congenital malformations, multifactorial diseases. Multifactorial is probably where epigenetics might have most impact. Mechanisms not necessary.
Makes me think that thank you. It's a basic question about the disease. If it's cancer biology. There's no transgenerational component. There's no radiation component. What is meant here? Is it are we still talking about in the progeny of irradiated parents? But maybe it's premature to talk about that because there is no evidence of any disease. Huh?
I think that there's quite a bit of anecdotal evidence, as it were, for congenital malformations, and then weak evidence backed up by the recent reappraisal of the ABOM data. So neural tube defects and some other chromosomal disorders are documented. So so I think our starting point is that there are some observations of these of these diseases or disorders. Yeah, so I'm just responding to you saying that there is no human evidence. So I think there is. Yeah.
Yeah, but I think I agree with Mitri that it's just premature. Although I would tend to say Mendelian disease, just no effect of. I mean, but I would suggest that for Mendelian diseases, which are really due to knockout of one gene, then either you have it's there or it's not there, and yeah, that's really genetic. Yeah, yeah. But otherwise.
But yeah, but chromosomal pregnancy.
No, I would think that for A and C, probably there are observational studies documented, right? But B and D probably very difficult, because even even if there is a change in chromosomes, for example, after radiation exposure, you have translocations, but that is not considered as chromosomal disorder.
So there's some evidence of Downs, increases in Downs syndrome.
Okay. Yeah. No, I think it is. No, because that's what I mentioned. Yeah. Yeah, that's why, because no, that's why, because we don't call them as chromosomal disorder. Yeah, yeah. I mean, I know you in the exactly in the morning you meant you, or I don't know, someone mentioned about clonal transmission, right? I know that in Chernobyl, somebody was studying about the clonal translocation in patient. I mean, yeah, you were, and I mean, I don't know.
So Prakash, what you're saying is that you would see if there was a classic translocation that was then viable and in a gamete, and then you've got a stable, balanced translocation, constitutional in that offspring, in that child. But that doesn't relate to chromosomal disorder. Yeah. The only live births that are chromosomal disorders are numerical or sex chromosome ones, really.
No, I was reviewing. I don't know who the author was, but it's a case study from Chernobyl. I can't remember the name. This was submitted to Mutation Research and it's confidential, right? I was review. Yeah, mechanistic. So for example, it is not a transgenerational study, and this particular scientist, he or she found a clonal translocation. And I was asking, if you would have studied, I mean, observed the clonal translocation, only say few years after the accident, then why it was not, or why the disease prediction was not made at that time. If the translocation is on a chromosome, which eventually will lead to, say, CML or AML type of chromosomal disorder.
I think that actually this is now mechanistic approaches. So we should take this question more from the point of view of judgment, if it's possible for Mendelian diseases to be to arise via genetic or epigenetic mechanisms, obviously genetic. Chromosomal disorders, I would vote for the genetic rather than epigenetic. Congenital malformations, it could be both. And for multifactorial, the epigenetic might be even more probable. But if it's, I think that just to take it from the point of view, if it would be possible that this kind of mechanism caused these diseases. Not very.
What we discussed today's morning, it's important to link the genetic, epigenetic endpoints with the diseases, and then using the mathematical modeling that could provide an answer on the relative contribution to the specific disease, especially for multifactorial. I mean, I would see it in this way. Maybe I'm not sure if I could make my idea clear, but if we have the measured genetic and epigenetic parameters in one study, and we can link with the outcomes, diseases in this study group, that would provide an opportunity to assess the contribution of each component, especially that's for multifactorial diseases. Do you think it makes sense?
Whether these clonal translocations are seen in all, sorry, I mean, I think it's okay. The clonal translocations are observed in all cell types?
I think we're missing. I'm getting confused. A clonal outgrowth, something that's associated with cancer, that's in a subset. Okay, but what we're talking about here is constitutional, what you're born with, isn't it? So it's a different point. So in terms of chromosomal disorders that result in live births that go beyond a few years. It's Downs, it trisomy 13, 18, but then they die quite young, microdeletion syndromes, and sex chromosome disorders. And really, they're the sort of known classic or known. So we can look at them quite straight, unless there's microdeletion syndromes that we're not aware of. But I think that if you go into is it Mittelman, there's a whole catalogue of what actually, and then imprinting is Patau's, there's the classic Angelman's and Patau's disease. So, but we don't know if there's could there be other imprinted disorders? But I think we would have known about that by now with all of the clinical genetic studies. So that is a defined group of disorders, I would have thought.
Prakash, you already touched what I wanted to say, that a major difference between genetic versus epigenetic mechanisms is that genetic mechanisms propagate more or less in a clonal manner in the progeny, while epigenetics, they don't. So they act in a non-targeted manner as well. So they are not clonal changes and not necessarily in the progeny of affected cells. And of course, the combination of the two is also possible.
A little complication to this discussion. Ratio between genetic and epigenetic, I think they're interrelated. Example is human colon cancer. In human colon cancer, very often promoters of mismatch repair genes are hypermethylated, which leads to the shutdown, downregulation of the mismatch repair genes. And these cells, and it is stable modification, epigenetic modification, which leads to the mutator phenotype. Because these DNA repair genes are not working, they are genetically unstable, these cells. and they can acquire any kind of genetic mutations after that. During during embryogenesis, there is a huge methylation, DNA methylation reprogramming that's happening in the cell to make them basically, to erase the previous history of the parental organism. So if epigenetic reprogramming is not working properly, I can easily imagine that it will lead to some genetic changes which can become clonal and become, the de novo mutation. So there is a potential. I'm not saying it can happen, but
we should not forget one thing, that first, if we are speaking in terms of AOP or using terms used in AOP, so the initiating event is radiation, and there is an early key event which modulates secretion of production of microRNAs. But microRNAs are secreted outside the cells, and they are taken up by other cells, so they can modulate cells which did not receive the initiating event. And this is, I guess, really important. And another thing is, which well, I consider it, maybe it's important, that if you take a look at the mechanisms what influence, for instance, the dynamics of microRNA production and microRNA secretion, then you end up and see that very many proteins involved in DNA damage, in fact, also contribute in regulating the secretory activity of the cells. And then there are proteins which shuttle between the nucleus and cytoplasm, depending on when to repair the damage and when to act as secretory regulators. And this is quite highly influenced by radiation. But the main thing what I want to say is that if we think that non-coding small RNAs are epigenetic factors, and they are, then methylation cannot be secreted outside the cells, histone modifications neither, but microRNAs can, and they can directly influence surrounding or even distal cells. And I think that matters.
So are the epigenetic effects non-targeted effects? Do we talk about actually about bystander effects and genomic instability? And then about the dose response, it might be very different, up and level.
No, I'm not sure whether we should go to bystander. Yeah, yeah. I was having this question whether mosaicism comes under Mendelian or chromosomal disorders.
I guess I'm coming. Yeah, but I guess maybe it's getting late in the day for me, but I'm really struggling with how are we trying. Surely we want to just ask this question. We can't answer this because, and say that it's a relevant question to explore, but that there are evidences of say congenital malformations, but we actually don't have the answers now to be able to say what's contributed to that, but maybe we want to explore that. Or
yes, well, and then in the, let's say, radiation protection or different protective guidelines, sometimes you have to do expert judgment. So you don't necessarily have the information, but you use the, for example, in this case, the general knowledge of genome and how it functions. the general knowledge on human genetics, and you can do some judgments. I don't know if it's helpful, but I think that in principle, if you look at because these are the classes of hereditary effects that are usually listed. So, and if they can be act via genetic or epigenetic mechanisms of both.
Perhaps we are not answering the question, because the question, I think, is not about what is the relative contribution. The question is, how can we study this? And then, yeah, I guess we can only do this with animal models, I think, and then, you know, combination effects like radiation plus some factor that we know modulates, modifies the epigenome, and then we see. I guess this is how we can study it. Because we will not provide an answer because we don't know.
Yeah. It was mentioned a few times that the epigenetic effects would be reversible. Would this also be somewhere where you would like to take that into account? Especially the rate at which they are reversed?
Sorry? Yeah, I guess so. I think you could also approach this in epidemiological studies, but of course, you can't control them, but you can take into account factors, exposure to some factors, if you have good cohorts, something that can be looked at.
We have a question here whether they would like us to join Group B. Or they can join us. Yes, and we can open the doors.
Okay, but if we can just open this, it'll be cooler. Yeah. Can you open this? Okay.
Okay.
We're done, and we don't know, we just want to find out. We want to learn from you anyway.
Do you have enough space to sit?
Let's try to open that. So
He can open, but he has to close again.
oh. Yes, and quickly.
Yeah, I think that the 20 is almost we covered in the previous two sessions.
Right. Well done. Okay, considering the severity of effects or detriment, do underlying mechanisms play a role? Most likely.
Yeah. Yeah. I think it's bigger to 19, right?
You're not sure.
I'm not sure I understand what this question is getting to.
It's asking about mechanisms.
Is this asking if
No,
there is a dose
severity of effects.
I don't get them.
Severity in detriment we don't have the right? Yeah, very severe.
If something happens, there is a mechanism for it.
Yeah.
I don't understand the question.
It's quite, I don't think it's
Not relevant. Really?
Yeah.
It wouldn't surprise me if it was asking if there is if we expect a dose response for all of these. Okay. Oh. Let's forget this one. Yeah. I think we have enough to discuss without it.
The last one is based on all we have been discussing.
After?
Yeah.
Not see?
No, but I think that here the question is, are there any interactions that are multiplicative? Because if they are, then by reducing your background level, you can reduce the radiation effect. So if you think, I mean, and for most cancers at least, we know that we assume the models are multiplicative, which means after radiation exposure, if you adopt a healthy life, and you reduce your background, you can reduce your radiation-induced risk. So I think you can. I would answer the question affirmatively and say yes, of course. If you change your lifestyle so that you reduce the additional genetic and epigenetic effects. Yeah, yeah, I know exactly. So it's a good advice.
I think it's a perfect advice, Sanjay, but I think it's not a radiation protection action to recommend a change in lifestyle. Is it? Okay. But radiation protection is about regulation. So you regulate, but you cannot regulate human habits. It's their choices. Yeah. So it's the way it's written, it's like actions. So it's like it's a prescription. It's like from the formal point of view.
Stop drinking, alcohol. I'm not looking at you now. Wait this long before you consume. That's people for radiotherapy. That's routinely recommended.
Recommendations, not actions.
They assume that late stage germ cells are more sensitive than.
But I don't think we have any, other than not having a child, any suggestions.
Yeah. So we have gone through all the questions now, and now
Now we go to the gold room.
the gold room.
Yes. Yeah. So tomorrow we talk about the structure on the paper. Yeah. And maybe about our. I'll go over my notes.
Are they opening or?
I'm also saying, Cisco, I have to leave slightly early tomorrow, but I think I should be okay to still be here for the presentation of our results. But I won't. I'll miss the end of the meeting. I think because I'm already having the last flight of the day home. Yeah. And I need to be back on Friday.
Collaborator asking, do you have an idea where we should go for the cruise? I don't know.
I like that question better than some of this.
We can have that as question 23.
No.
The
two
Europa
people
dinner.
up here. They resisted. I didn't resist them. So, yeah, but they don't know what to do.
I don't know. I don't have a program. It's Europa event.
Yeah, no, this year, yeah. Well, I'm going to ask them.
Yeah.
There is a link. Okay. John and Svetlana, we are joining Group B now. If you have the link, you can log into that group B session. Svetlana, okay?
Thank you.
Yeah,
Thank you.
thank you. Yeah,
Okay. Thank you.
yeah,
Thank you.
good. Thank you.